special in its makeup. Dr. Herculano initially was trained as a biologist at la Universidad Federal de Rio de Janeiro in Brazil. And later she followed a master's degree in, is she here? No. At Case Western with Dr. Story Landis, followed by a PhD at the Université Pierre and Marie Curie in France. Dr. Herculano is an author of The Human Advantage, a book that is quite popular, in which she tells the story of her discoveries on how many neurons different species have and how the numbers of neurons in the cerebral cortex of humans is the largest of them all, thanks to the calories amassed by the very early technology developed by our ancestors, which is cooking. And I have to say, Dr. Herculano is a wonderful communicator. I follow her in her TED Talks. Specifically, I use her TED Talk from 2015 in my neuroscience courses. So it is a pleasure to have her, and I hope we can enjoy her presentation despite the difficulties.
All right, thank you, Carmen, so much. Thank you all for hanging in there and joining this second meeting now. I hope this goes okay.
I don't see the presentation, though.
You can't see the presentation again?
No.
Google keeps kicking me out then.
Oh. Let's try one more time, and if not, we'll cancel.
I have my microphone, I have my camera. Present screen again.
People says that they cannot hear anything. That's strange.
Okay.
Go again. There.
Do you see my screen?
Yes, ma'am. So let's start.
Okay, let me try again then. So without further ado, I'll tell you about work that I've been doing and where it's headed now on what you see illustrated on the screen, I hope, which is my main topic of interest, and that's biological diversity. Like Carmen said, I'm a biologist by training. I later became specialized in neuroscience, and I'm now interested in understanding not only how diversity happens, especially in the case of the nervous system, but also what difference does it make? What does it matter that brains come in different sizes and are possibly made of different numbers of neurons? So when I first became interested in diversity, there was a consensus of sorts in the field, in that most people were content to suppose that all mammalian brains were built the same way. And by built the same way, that they meant that if you had two brains of a similar size, they would be made of similar numbers of neurons. And if you had a brain that was much larger than another, say an elephant brain, which is the size of my forearm, it's about 3 times as large as a human brain. That larger brain should
Suzana,
have more neurons.
Oh. Suzana?
Yeah, did you lose the presentation?
Suzana, again? You're not. Yeah.
Why don't we, Dr. Herculano, why don't you just give like you would do your TED Talk, but you can see the presentation, but at least we see you. So is that a possibility?
Yeah, it's just that somehow you see me, but it says I got kicked out from the meeting. Just a second.
Now we see.
Can you see me?
Yes, ma'am.
Okay. All right. Yeah, I can definitely do that. So forget about my presentation. I'll just talk to you guys. So what I'm interested in is diversity. What are different brains made of? How does that happen? How do different brains come into existence? What is it that defines that one brain, like a mouse brain, it will be this tiny, and an elephant brain can be enormous, 3 times the size of ours. And initially, the expectation was that, well, all that we're looking at there is really just a variation in size, because the rules, let's say, of behind how these different brains are built, they were supposed to be the same. And what that means is that if you had 2 brains of a similar size, then you would expect those 2 brains to be made of similar numbers of neurons. And if you had one brain that's much bigger than another, and this is important to us because our human brain is not the largest brain around, then that larger brain, like an elephant brain, should have more neurons than that smaller brain, like the human brain, which is a very reasonable expectation to have. The problem is that, well, we run into these conundrums, like, for instance, the human brain not being the biggest one. So, and this is a conundrum indeed, because we like to think that the information processing units of brains are neurons. Some people, when I say this, they argue, no, but they should be the synapses. The synapse is the unit where signals are exchanged. That's true, but all the synaptic input onto one neuron is combined into that cell body, and there is the one input that is generated from the ensemble of all those synapses that are active or inactive at any moment in time. So the amount of signal, the number of signaling units that you have in a brain should be really considered the number of neurons that it has, and not the number of synapses. So the issue of how can humans have, well, how come we're the ones studying other animals and not the other way around? How come it's not elephants studying us? Since they have the bigger brain, so supposedly they have more neurons, especially in the cortex, that top part of the brain that has control, that has the possibility of modifying all circuits, circuit activity in other parts of the brain. And so I started looking for the numbers, because back then, this is 15 years ago, but until the latest, the one before the latest edition of Condell, Jessel, and Schwartz's Principles of Neuroscience, you would find in that the human brain has 100 billion neurons and 10 times as many glial cells, period. That's it. So I wondered where that came from. Do we really know that? Or is this just an order of estimate, an order of magnitude estimate, and we actually don't know what different brains are made of and how numbers compare? And long story short, that I could never find the actual evidence for of numbers of neurons that compose different species. We actually didn't have, we didn't know how many neurons human brains were made of. We did not know whether elephant brains actually had more neurons than human brains, or if they had similar numbers or even fewer. That number just did not exist. Same for comparisons between mice and rats. There were no direct estimates. And the reason I realized was that, interestingly enough, there were no estimates because people thought that this was solved already. This is not a question. This is not an issue anymore. We already know that, look, it says here in the textbook that we have 100 billion neurons and 10 times as many glial cells in the human brain. So that was problem number one. Everybody thought that this was solved already. We know that bigger brains have more neurons. We know that 2 brains of the same size, like a chimpanzee and a cow, they have brains of about the same size. They're about this big. And yet a chimpanzee behaves like a chimpanzee does, and the cow is content to just spend the day just grazing away. So they obviously have very different behavioral repertoires and behavioral capabilities. So it seemed to me like it was a worthwhile question. How many neurons and how many glial cells are different brains actually made of? And once we have those numbers for a good range of species, what story would those numbers tell us about scaling, about diversity, and about how the human brain relates to others? So I had to come up with a method for that, because that was problem number two. The method for estimating numbers of cells that compose different tissues until then was limited to stereology, which means that you need to have the full tissue, you have to have it fully sectioned, you have to have it fully treated for histology, you have to use a very, very precise sampling method. And then your estimate in the end is still going to be dependent on the volume that you sampled, even if you use some mathematical expedients to try to do away with that. So the way I worked out to figure out how many neurons different brains are made of was pretty much to turn those brains into soup. So you take fixed tissue, you break that tissue down into the regions that interest you. So for instance, we always work with the cerebral cortex, separate from the cerebellum, separate from the other parts of the brainstem and thalamus and the basal nuclei. Anything that you can dissect, you can count. And that way you, instead of looking at the tissue, what you look at is the dissociated cell nuclei, because the process of turning the tissue into soup is essentially the process of dissolving, of literally liquefying the tissue in detergent. The detergent dissolves the cell membranes and releases all the nuclei from the cells. What that means is that as long as it remains true that every cell has one and only one cell nucleus, then we don't need to count cells anymore. We can count cell nuclei. And that's a huge difference, because whole cells are distributed very unevenly in the tissue, so counting them becomes very difficult. But when you dissolve the tissue and all you have is this suspension, this liquid that pools all the cell nuclei that once were in the tissue, now you have a very straightforward way to count them by sampling under a microscope. So you can use a counting chamber, and in 10 or 15 minutes, you can get 4 or 5 direct counts of how many cells you have, how many nuclei you have per small volume of that suspension. And therefore, since you know the total volume of the suspension that you turn that brain or brain area into, you can estimate how many cells and then how many neurons by using neuron-specific markers were in that tissue. And the main takeaway, after having looked at a few dozen species and several individuals of many of those species, is that there is an enormous range of diversity in how different brains are made. And so the bottom line is that even if it's often the case that if you take a bigger brain and a smaller brain, the bigger brain does have more neurons, that is not always the case. And there are consistencies in those differences. So for instance, we found that compared to other mammals, primates have many more neurons packed into a brain of the same size. So if you take a primate and a non-primate mammalian brain, you find that the primate has many more neurons in that same brain volume. And even more than primates, birds have even more neurons hiding in that same, even relatively small brain. So the difference lies in what happens to the average size of the neurons as different species gain different numbers of neurons in evolution. And that remains one, I think, very important open question. We do not understand what is it that controls, that regulates how large the average neuron is in a brain. We can estimate now from our numbers that cortical neurons in the cerebral cortex of an elephant are about 40 times as large in volume, in the volume of the average cell. And I do not mean just the cell body. I'm talking about the cell body plus all the axons and dendrites. So there's this 40-fold variation in the size of the neurons. And this is really important to, and also to put into context, because I found that when I talk to specialists in neuroscience, we're very used, we accept very well this idea that, well, yes, of course, an elephant neuron must be bigger than than a human neuron, right? Except that no, that's a very unreasonable expectation, because if you compare other types of cells in the body, and we're doing that ourselves now to have our own data acquired in it the same way as we do with neurons, you realize that, for instance, the average size of the cells in the liver or in the kidneys or in the lungs of an elephant, a mouse, a giraffe, a human, or a monkey, they're all the same. So it's really there's something about the neurons that allows the size of the average size of the the size of the average neuronal cell to vary a lot. So that is where most of this diversity lies. And you have diversity in the number of neurons that compose a structure, and you have diversity in how that number relates to the average size of the cell. So that's how you end up with a bird, a primate, and a non-primate mammal that have brains of a similar size. And what you're going to find is that the primate may have 5 times as many neurons as the non-primate mammal, and the bird might actually still have 3 or 4 times as many neurons as the primate itself. So let me summarize it this way. Yes, primates are different compared to other mammalian species, and we are one of those primates. So we are no different from what you would expect a generic primate of our size or our brain size to be. We have that number of neurons, which, by the way, is not 100 billion and 10 times as many glial cells. It's about 86 billion and just as many non-neuronal cells, which is very important, because those are the numbers that you would expect for the brain of a generic primate of our brain size. So so there are a number of interesting questions to pursue there. For instance, how does that variation happen? How does that relate to the different shape of the cortex that you have in different species? We found that, for instance, the degree of folding of the cortex is not as it was expected. It's not simply a function of the size of the brain, and it's also not a function of how many neurons that cortex has. What it is the physical, it's the mathematical function that you would expect from the pure physics of volume, that as it expands in development, it has what's the name? It has a self-avoiding surface, meaning as that volume gets folded or crumped under pressure, its surfaces bend and even fold, even touch, but they don't fuse. So they're self-avoiding. So it's really interesting that once we could model from first principles the physics of how a cortex of a certain surface area and a certain thickness, how much that should fold once it forms under these constraints, we can predict, turns out, 99.8% of the variation in the degree of folding of different brains, which in biology is really spectacular. We usually satisfy ourselves with 30% or 40% of variation explained, which led me to the other issues of what are the implications of having a brain that that has this same number of neurons, and yet one is very large and the other is very small, or having brains that are the same size but actually have completely different numbers of neurons. So the usual expectation, what people usually associate with variations in numbers of neurons, is cognitive capabilities. It's what can you do? What are you capable of doing with the number of neurons that you have? And in the case of the cerebral cortex in particular, since the cortex is this part of the brain that receives copies of all the information that is processed in the other structures, and it has all this intrinsic connectivity, this associative connectivity that allows for new combinations of signals to be created. You have new associations. The cerebral cortex really is the part of the brain that contributes to making behavior flexible and complex. And flexibility that also leads to complexity is something that I've come to associate with intelligence. That's another story that I could tell you, but for now it'll suffices to say that the more neurons you have in a cerebral cortex, the more capable that cortex should be of creating new associations, of modifying behavior, of making behavior more flexible and therefore also more complex. And that is very likely something that, at least one of the things that we call intelligence when we're looking at the behavior of different animals. So that, of course, is a huge, is an enormous consequence. The more neurons a species has, the more cognitively capable you would expect it to be. And it's highly satisfying because there, for the first time, we have a clear cut feature that separates our species from everybody else. So to go back to the elephant, we now know that even though the cortex of the elephant is twice the volume of a human cortex, ours actually has 3 times as many neurons as the elephant cortex. So we have on average 16 billion neurons in the human cerebral cortex. In the elephant, you have barely 5.5. So that's definitely very satisfying that we find that there is something remarkable about the human brain. We do have the most neurons in the cerebral cortex, even if we don't have the largest cerebral cortex. And no, that's not because there's something absolutely outstanding or out of the ordinary or special or unexpected about what human brains are made of. We have the cellular makeup that you would expect for a generic primate brain the size of ours. So what's really so important about, what's really so remarkable about us is that we are primates, so our neurons are on average fairly small. And on top of that, we are the primate species that managed to have the most neurons in the cortex, which, and this is where you should start thinking of the other implications of having varying numbers of neurons. We know that brains are very expensive. We still don't understand what exactly determines exactly how expensive a brain is, meaning how much energy a brain costs. We do know, however, that it seems to be a linear function. So it's just a simple proportionality of how many neurons you have in that brain. So if you have 10 times more neurons in the brain, you can expect that brain to cost 10 times as much energy, whatever size it actually has. So that's one of the things that we're working on right now, is figuring out how is that established, what determines how much energy a brain costs. But that's really important and consequential, because when we're talking about human evolution, and if you look at those images of human brain evolution, what's so striking about the evolution of the human brain is how enormous that the human brain became in a relatively short amount of time, which is 2 million years, which is still, of course, a very, very long time. But in evolutionary time, we see that in 15 million years, primate brains go from very small to about this size in 50 million years. And they go from this to the size of a human brain in just 2. So I became suspicious that their energy must be a limitation. Maybe once you start having that many neurons, it's actually very hard and unlikely for a brain to be able to, for a body to be able to eat enough to sustain both that body that's becoming larger and a larger brain with larger numbers of neurons. And once we were able to do the math, that's exactly what we found out, that if we ate like other primate species do, if we humans had the same diet that our fellow great apes have, we would have to spend 9.5 hours every day looking for food and eating. So imagine what your day would be like if you had to spend 9.5 hours of it every single day, just looking for food, preparing that food, and eating that food. We wouldn't be here. There wouldn't be enough time in the day. There wouldn't be enough hours in the day to do all the things that we think are part of regular life these days. And actually, there probably wouldn't even be enough time, enough waking hours for us to get enough food to begin with. If we look at other primates, we know that orangutans and gorillas, when eating 8 hours, 8 solid hours every day, is no longer enough because it's the rainy season or there's a drought and there's food is scarce, the animals lose weight. So 8 hours of eating might not be enough, and it looks like more than that is actually not possible, not feasible for a primate, because remember, we sleep between 6 and 8 hours, and that's something that all primates do. So there's that consequence as well. And if you're now wondering, then if we should eat 9.5 hours per day and primates can't do that, then you're saying that we shouldn't be here. And that's exactly what I'm saying, that if we ate like other primate species do even nowadays, we would not be viable. Our species, with the size of our body and the size of the brain, with the number of neurons that we have in the brain, would not be viable. And the fact that we obviously are here is evidence that that problem got solved somewhere in the past by our ancestors. And according to our math, the only way to solve that problem, if you would have to eat 9.5 hours, that's not enough. You can't even do 8. And the human brain actually it still costs just as much energy as you would expect for the number of neurons that it has, is that the only way out of that conundrum is that our ancestors must have come up with some kind of cheat. They must have found a way to increase the amount of energy that they could get out of the same foods that remained available. And it turns out that we know that cheat, or we know one such cheat that is enormously helpful and important in modern life, so much so that we're able to take it for granted. And that's cooking. It's become such an important part of everyday modern life that we no longer even think about it. We don't pause to consider that it once wasn't there, and other animals do not modify the food that they eat as much as we do. So there's a lot of I like to think of cooking as one very early technology that our ancestors developed. And one very consequential technology because it was one that allowed our ancestors to afford a brain with an unprecedented number of cortical neurons. Now, the main story that I planned on telling you today is yet another consequence of, well, in that sense, another consequence of this technological innovation that was cooking, which allowed us more numbers of neurons. And that is the pace of life, how long it takes different animals, different warm-blooded animals is what I'm talking about. How long does it take them to develop? And once their body matures, once they reach sexual maturity and they become capable of reproducing, how long do different animal species live? And this is a question that I of course I'm biased, but I think that it should be considered much more important than it is, because we're really looking at extremely consequential biological diversity. The question here is, how come a mouse, not only how come a mouse is mouse sized and an elephant is elephant sized, of course, but how come that mouse, it doesn't matter how well you treat it, how well you take care of it, that mouse will not live much longer than 2 years. But an elephant is still developing at 2 years of age. It's still not fully grown. And an elephant will actually live over 60 years. So there must be at some very fundamental level of differences across species and across warm-blooded species in particular, that allows for this enormous range of variation, where you have creatures that live between 1 and 2 years, and that's it. That's all the life that they're gonna have, which means that they develop very fast, they become reproductively competent very quickly, but they also live and die very fast. And yet you have species like us, where we're cute, but still pretty much useless at 2 years of age. Takes a long time to develop and mature, but we're alive for a long time after that. So the short version of what people used to think about this was that the main determinant was body size. So, and the logic here was that it seems that bigger bodies use energy more slowly per unit chunk of volume in the body. So 1 gram of elephant uses much less energy per unit time than 1 gram of mouse body. That's fairly well established, and it's a very, very nice trend. Where that comes from is a whole other issue that I think is really interesting too. And right now, the best guess is it has to do with the physics of the scaling of distribution systems, like the circulatory system. That's a whole other story. If you'd like to know more, I'd be happy to help you find material to read on that. But anyway, so the notion has been for a very long time that variations in life history and the pace of life have to do with the size of the body, because with different body sizes comes different rates of using energy. And just like we're comfortable with the idea that a strong fire burns faster, the idea here was that animals with faster metabolism, they should also accumulate damages much faster, and they should expire faster. They're going to reach that point where damages are too many to withstand, too many for the body to remain functional. that you simply collapse. You no longer function. So because metabolism slows down as warm-blooded animals become larger, then the consensus was that, well, it makes perfect sense then, and we can even do some math that seems to support this, that the bigger an animal is, the more slowly it uses energy, therefore the more slowly it accumulates damages, therefore the more slowly it's going to age, and thus the longer it's going to live. Now, what happens is that I found a database with all these numbers of longevity and how long it takes for a warm-blooded animal to develop into sexual maturity. And it turns out that, yes, there is a very obvious and very decent correlation with the size of the body. So that's a correlation with an R square. So a coefficient of correlation of about 0.34. What that means is that about 34% of the variation in longevity across warm-blooded species can be accounted for by the variation in the size of the body of those species, by variation in the mass of the body. That's decent enough, it's reasonable enough that you do find, when you look at this graph, you do see a correlation. You do see that the more, the larger the body is, the longer the animals tend to live. What you won't realize, unless you bother to color code the data according to clades, if you group the species, that's what it means. You won't realize that that correlation is not universal. And there's a very interesting pattern of segregation. There's a very interesting clustering behind that might sound familiar to you at this point, which is that when you compare a bird, a primate, and a non-primate mammalian species of a similar body size, you find that the bird lives much longer than the primate, which in turn lives much longer than the non-primate mammal, which is exactly the relationship that we had found between the size of the body or the size of the brain and the number of neurons. So for a similar body size or for a similar brain size, birds always have more neurons than the primates, which have more neurons than the non-primate mammals. So that made me very suspicious that could it be that simple, actually, that it's not the size of the body that matters, it's simply how many neurons you have in the brain. And it turned out that in the cortex in particular, that's what really seems to matter. The more neurons you find in the cortex of a warm-blooded species, and it doesn't matter if it's a bird or a mammal, the longer it takes that species to reach sexual maturity and the longer that species lives after having reached sexual maturity. And this is a correlation that not only it truly is universal, you have bird species lined up very tidily with primates, with all other mammalian species that we could look at, but also it's a much stronger correlation. So when we use numbers of neurons, regardless of the size of the brain or regardless of the size of the body, what we find is that you can account for 74 of the variation in longevity across species, simply by taking into consideration how many neurons you have in the cortex. So this is early days of this finding. So of course we would love to know first, is this simply a correlation? Is there actually a causative link between how many, between getting more neurons and being able to live longer, taking longer to mature and then to live? So that is work that we're setting up to do now in the upcoming years. But I wanted to wrap up by just putting just commenting on the significance of this new finding in the light of the story of how the human brain evolved. Because even if there's no causation here, even if the number of neurons in the brain is simply just a very good predictor by pure chance or whatever, but it still is. It is a very good predictor of time to mature and then lifespan, your maximal lifespan, that is. That means that in human evolution, during those last 2 million years in which the size of the human brain tripled, we can actually predict, we can actually do the math and estimate that the age at which human children reached sexual maturity has been increasing. So childhood has been becoming longer and longer, has been lengthening from 6 to 8 years to now 11 or 12. Just like maximal longevity, we can also estimate that also has been increasing from a maximal lifespan of 60 years or so to a maximum 90 to 100 years calculated for the number of neurons that we have nowadays in the cerebral cortex. If that doesn't sound like much, just consider what has been happening lately in the recent 2 centuries to the not maximal, the average longevity. We've seen how many people used to live 20 years, if at all. A 50-year-old person was very, very rare to find. And nowadays, that's not that rare anymore. Now the average longevity has increased. The average life expectancy is upwards of 70 years. So that is an enormous change that I'd like to wrap up by inviting everybody to consider the consequences of those changes, of that extension in human lifetime, not only in the evolution of our species, but also in what we now call the history of our species. Because I think what is left for us to realize from that is that when we compare our species to others, when we compare humans to mice, when we do the biological research, we work with this expectation that all it takes is just having a brain that's 1,000 times bigger than the other with more neurons, and that's it. You get more neurons, and along with the 1,000-fold more neurons, you get lots more cognitive capabilities. And it has to be much, much more than that. And I think that it's time for us to start considering that time aspect, that time dimension of how different species compare to each other, especially the human species to others, because if given that the more neurons you have in the cortex in evolution, the more life slows down and the longer it takes you to reach independence and the longer you will live after you've reached that independence, that means that not only with more neurons, you get more cognitive capabilities, you get more capabilities of using, of working in the information that you get, but you're also going to get more time. You're gonna gain more lifetime to develop under the supervision of your parents as a child. So human childhood is now upwards of 10 years. Nobody else, no other species has that long a period of time of opportunity to learn from their parents, to learn from their peers, while still not having to depend on their own on their own capabilities to remain alive or to function. And then after that, you have the possibility of you have your adult life, you have decades of adult life to solve problems, to create technologies that help you solve those problems even better and faster, which in turn now gives you even more free time that allows you to become interested in even more complicated problems. So technology is the solution to problems that allows us to find even more complicated problems to get busy with. And that's really important because that has been the history of our cognitive, not evolution, our cognitive progress, which is directly linked to the pace of life and how slowly or fast we live life. So the bottom line here is that not only the human species is not an outlier in how many neurons we have or how our brain is put together. We are a primate. We are one more primate. And as that one more primate, we have a number of neurons that is nevertheless very large. And along with that number of neurons comes, just as predicted, a longer childhood and a much longer adult life that, remember, no mouse is ever going to have. So I think this is something that is very important, not only for considering for the study of aging and longevity itself, because we've been using mice. Simply, we've accepted this idea of biological time that a 2-year-old mouse is equivalent to an 80-year-old human. We accept that a dog, a year for a dog amounts to about 6 or 7 years for a human. So we've bought into the idea that we can study 2-year-old mice as if they were the equivalent of very old humans. And if the consequences of time are if the biological consequences of time are simply proportional to time lived and how much damage is accumulated during that time, what we have here is that, of course, no other species will be able to live as long as we do and accumulate as much damage in their brains as we do. So it's no wonder, to me, in that scenario, it's no wonder that the mouse is a very imperfect model to study human aging. You cannot get most of the signs of the destruction that happens with Alzheimer's disease over 70 or 80 years of living in a human brain. You cannot get that in a mouse. So there are workarounds, possibly, once we understand what it is really about, the number of neurons in the brain that contributes, that determines how the speed at which life progresses, and also not only development and maturation, but also longevity. Once we understand that link, we should be able to gain not only fundamental information, fundamental insight on what is aging exactly, how is it that the more neurons a species has in the cortex, the more slowly it ages, the more slowly it develops also, but the more slowly it ages, and therefore the longer it can live. How does that happen? And also, once we have that insight, then we can hopefully start appreciating more the importance that our long lives have to our cognitive development and historical progress, really, because the sure, it's great that we are primates. We have the primate biological makeup of our brain, meaning that our brains are made of tiny little neurons. So there's plenty of them in a brain that's not all that large. But still, there's this whole other dimension of a very long lifetime that comes with that, and that is a lifetime of opportunities to learn and exchange ideas and learn from our parents, learn from our elders, then learn ourselves, learn from our peers, and then eventually we become ourselves the teachers. We become the older ones in the population whose role then it is to pass that information onwards. So I'll finish with this on this note that it's thanks to this very large number of neurons that we have in the cerebral cortex and the very slow and long life that came along with it, somehow came along with it, that what we're doing here, the opportunity to bring young and old and not so old together at the same venue, if not in the same meeting room or the same university, that is, to me, that's the essence of what has made us modern humans. It's what we have that builds on that makes much more than the difference between human biology and, say, mouse biology. It's not only the difference in numbers of neurons that we have, it's all the time that we gain to use those neurons and develop technologies and generate complexity and flexibility in our lives. All that thanks to this one thing that we still have no good idea how it works, and that is the diversity in biological time. So I'll stop here, and hopefully that still leaves Bruce some time and some time for questions. So thank you all for being there.
I want to thank you. You gave a superb presentation under very difficult circumstances, much better than anything we have seen with slides and movies and everything. So I really want to thank you. And I have two questions. You touched about the glia, but didn't develop it. Do the ratio that you see with glia and neurons stand between all the animal species? And my second question has to do with birds, because
So
birds,
can I
there
answer the first?
Sure.
Let me answer the first one. So this is very interesting that at the same time that neurons come in all sorts of sizes and shapes, glial cells are like any other cell type in the body. There's a certain average size of the glial cells in any species. There's a little bit of variation, and by little bit of variation, I'm talking about maybe 2 or 3 times variation around that average, but it's not 1,000 times variation like you have in neurons. So what this means is that when you take that chimpanzee and a cow brain that have the same size, they do have similar numbers of glial cells. When you take the elephant cortex, that's twice the size of a human cortex, the elephant cortex does have twice the number of glial cells that we have.
Fascinating. My other question has to do with birds. You know, like there are intelligent birds and sort of not so intelligent birds. If you compare the size, you know, let's say you have 2 birds with similar brain size. Will the birds that are supposed to be more intelligent, like crows or ravens, will they have more neurons?
They do. They do. So the diversity that we see across mammalian clades is also there in birds. We had a first set of 28 bird species, and you find that songbirds, including crows, and the parrots, they're the ones that have the most neurons in the brain, especially in the pallium, which is essentially the bird version of the cerebral cortex. And there's more data coming up now from my collaborator in the Czech Republic, Pavel Němec. He's the one carrying on the work with literally dozens of bird species. And that's exactly what you see, that there are different groups of birds that just like primates and say rodents that show that difference, where for a similar brain size, songbirds and crows have many more neurons in their pallium than say ducks or chicken.
Thank you very much.
Wonderful. I had a question, because what you brought up about how can now extrapolate what we see in a mouse into human biology, right? You just mentioned to us that this is something that we will have to be very careful of.
Yes.
Making these assumptions that we are having two brains that are working the same. But then, since I work with animal models, and I use the rat for addressing questions that could relate to translational research. How do you approach that from the standpoint of telling us that we are using animal models to answer human questions, if we are, obviously we cannot, for obvious, in my opinion, obvious reasons, to experiment with the human brain. Nonetheless, what is your opinion on that in particular?
So I let me say something positive here already. I think there is a way out, and that's exactly that's one of the reasons why it's so important to understand what is aging and how does that happen, and what is it that happens that is related to how long you get to live. So that's longevity. And there are two main suspects right now, and one is the changes that happen to proteostasis with time. So just the capacity of the body to keep its proteins well folded and literally in good shape, and which, so protein malfunction, protein malfolding, misfolding accumulates over time. The other thing that accumulates over time and seems to be very, very limiting is DNA damages. So there are known animal models of progeria. Progeria is that terrible genetic disease where individuals age incredibly fast, as in you find a 10 or 12-year-old young human who actually already has shriveled skin and the hair has gone gray, and there's signs of organ malfunction throughout the body. So we know now that progeria is a disorder of abnormal DNA damage and accumulation. And mouse models that have those problems with repair of DNA damages, they have accelerated aging, and they show much worse. Their brain looks much older than the age that they actually are. So I think it's going to be very likely that we can learn to turn to those models of accelerated aging to make a mouse model or a rat model much more equivalent to an aging monkey or even an aging human. If once we use that as a tool to get a level of damage accumulated in a short-lived creature that would otherwise never ever happen.
Mm-hmm. That's very interesting. Wonderful.
Actually, if I could make a comment, Suzana.
Yeah, Bruce.
So elephants actually have 20 copies of P53, which is an enzyme very important for a transcription factor, very important for DNA repair.
Right.
And I wanted to ask a question, if it's okay, since we're at the same institution. But what about naked mole rats? We don't even know how long they live. It's estimated they live beyond 30 years.
They do. So let me just first say something about the elephant. So what we've been doing so far, Bruce, is just hanging on to these one difference that seems to support what you're looking at, right? So elephant is super long lived and it has all these copies of P53. Yeah, it also has a number of other things, but when you look at how the elephant compares to other species with its 20 copies of P53, it still lives exactly as long as you would predict for its number of cortical neurons. So that's to say, it's sure maybe the extra copies of P53 help. They definitely help them not get as many tumors. I would expect, but it's still it is out of the ordinary in a sense, but it's not that out of the ordinary that it makes the elephant an outlier in how long it lives. See, now the naked mole rat, there's an interesting added aspect here. I've been insisting on warm-blooded animals. Warm-blooded animals are animals that function between 37 and 39 Celsius. And that's really important because that proteostasis thing that I was talking about, it's directly dependent on absolute temperature. And naked mole rats and they're actually not the only ones. There are other subterranean rodents that actually function at much lower body temperatures. The naked mole rat functions at 34, and it can actually have a temperature, an even bigger drop in body temperature. And so it's very, and we know that other bizarrely long-lived warm-blooded animals like temperate climate bats, they hibernate. So they also don't follow the rules that apply to all the other warm-blooded animals. So it's very possible that the exact body temperature, or at least whether you function in the 37 to 39 Celsius range, or whether you function below that probably plays an enormous role in pace of life.
Hey, that is what I am, it's so interesting. I learned so much about
Thank you. My pleasure.
Yeah, it was amazing. I know some students stayed for as well to have any questions to Dr. Carter. I mean, is there any students want to have a question to Dr. Carter about the Vanderbilt program? You can unmute yourself, so make it quicker than writing it. Any questions? No? Come on, guys. Okay.
Maybe I just covered everything so clearly when I presented.
Well, sounds like it. But now, nevertheless, we have all the information from Vanderbilt, not only through the PowerPoint presentation that Dr. Carter offered. We also have their links and obviously emails from make it Dr. Herculano or Dr. Carter for any questions you may have. You can reach us or go straight to them, obviously, and say, that you were here and that in the seminar series. So I think that will also work, of course. So I would like to once again thank both Dr. Herculano and Dr. Carter, especially Dr. Herculano. I am so appreciative of your patience. I don't know what happened, but
Don't worry, my pleasure.
Did it. and which was our aim. And we had a wonderful talk, and I really thank you for what you have shared with our students and with us. And then we hope that you have continue to find this new, very important knowledge about the brain and the evolution of the brain, evolution of us as species. And I have to tell you that I'm a fan of yours every time I get to see you talking at
Thank you.
So thank you all for being here. And if you have time, of course, you can join us next week so we can have another seminar. So buenas noches, good night, and
Good night.
take care. Bye.
Bye-bye.